Hey guys, in this video we're going to learn some of the terminology we're going to use to refer to DNA. So I'm sharing my screen with you here, and you'll be able to see our slides from chapter 10, which are on Blackboard. And you'll see that our objective for this video is to learn about these two terms we use to describe DNA, which is that DNA has a 3' end and a 5' end. So we have to see what that means. So remember, DNA molecules contain the sugar called deoxyribose. And if you write out the chemical formula for that sugar, there are 5 carbons, C5, there are 10 hydrogens, H10, and then there are 4 oxygens, because remember, 1 oxygen's missing. That's why it's called deoxyribose. So if you look at the structure of the deoxyribose, in the center here, there's an oxygen, but then the carbons can be numbered, 1, 2, 3, 4, and 5. So everywhere there's a bend in the structure here, that's where a carbon is located. So here in this corner, that's carbon 1. Here in this corner, that's carbon 2. Here in this corner, that's carbon 3. Here in this corner, that's carbon 4. And then carbon 5 is out on this little stick to the side. So we're going to see that the phosphates that link the sugars together connect to the 3 carbon and the 5 carbon. So let's see how that works. So our deoxyribose is going there, and then you can see how other sugars and phosphates link onto that first deoxyribose. So here's a deoxyribose sugar, and notice that the phosphate is linked onto that carbon number 5 here, and then the phosphate links onto carbon number 3 of the one above. So we've got a 5 and a 3 and a 5 and a 3 and a 5 and a 3. Now notice up this end of the molecule, the 5' carbon is free, right? And down this end of the molecule right here, the 3' carbon is free. So we say that DNA has ends, 3' end and 5' end. So here's our sugars phosphate, sugar phosphate. So we would refer to this end where the number 3 carbon is open and something could bind to it. We would refer to this end as the 3' end of the DNA. And then down the other end where we've got the stick sticking out and the 5' carbon is available, we'd refer to that as the 5' end of your DNA. So you can see that we've got a 3' end and a 5' end to our DNA. So if you look at the DNA molecule with the hydrogen bonds holding the 2 strands of DNA together, you'll notice that the strands are called anti-parallel. So that means that this one is kind of going this way, 3' to 5', and this one is going this way, 3' to 5'. So you'll see that the strands are going kind of opposite directions. You'll also see that between A and T, you've got your double hydrogen bonds, and between G and C, you've got your triple hydrogen bonds. And remember, you're always going to have a purine bound to a pyrimidine. The purines are the 2-ring structures. The pyrimidines are the 1-ring structures. So in your DNA, this terminology, 3' or 5', it tells you which end of the molecule is open. So at the 5' end, I've got an open number 5 carbon there that something could link onto. At the 3' end, I've got an open number 3 carbon there that something could link onto. So we're going to use this terminology 5', 3', when we're talking about the direction that enzyme moves along our DNA molecule to read the bases and to make copies of DNA. So that's our the end of this short video about the terminology used to describe the ends of the DNA.